Works. This is just a topic overview so that when you're revising, you're aware of all the things that you need to know, as opposed to just thinking to yourself, oh yeah, I know how respiration works. Yes, you might know how respiration works, but do you know all the stuff that is in the topic of respiration? Because it would be a bit of a shame, for example, if you knew how respiration worked, but you forgot to revise how respiration can be measured, or that you knew how respiration works, or aerobic respiration works, but you forgot to kind of polish up the bits on anaerobic respiration. Okay, so we're just going to make sure that you guys are aware of what all the things that are in the topic. And we're going to do that in a map form. Okay, so I've made a start here because usually what happens is I make a start, I put things in the wrong place, and then it gets a bit awkward for me. So made a bit of a start here. So we start with respiration, and big picture is always first. We start with why respiration is needed. Okay, so respiration is needed because respiration produces ATP. Okay, respiration, in our previous discussion about photosynthesis, we discussed how respiration and photosynthesis are kind of equal and opposite processes. While respiration takes inorganic carbon dioxide, simple inorganic molecule, combines it using the energy of light, so puts energy in to produce this more complex organic molecule made of carbon, essentially storing energy. Respiration is the opposite process, where we're going to take a more complex organic molecule like glucose, break it down back to CO2, carbon dioxide, an inorganic simple molecule, and in that process release energy, right? And that energy, when it's not just released into the cell, it's actually used to make ATP. So that energy released is stored as ATP, and then whenever there's a process in the cell that needs energy to happen, the ATP is hydrolyzed at that point. We're saying that glucose broken down to CO2, energy is released, but that energy is used to convert ADP into ATP. And then when we go back in that process, energy can be released to drive many processes. Now, reading between the lines, in your biology course, you've looked at a lot of processes that needed energy, and this is the time when you try and put that all together. So what is that energy used for? Well, the most obvious answer is active transport. We also have anabolic reactions, any synthesis process. Anytime where we have to take something simple like amino acids and build them up to something that's more complex like proteins, any time where we started with monosaccharides and we went to polysaccharides, any time where we went from simple to complex, small to large, that requires bond formation, and that requires energy input. Okay, so anabolic reactions such as, we can just name a few, like glycogenesis, the formation of glycogen from glucose. Other anabolic processes might include transcription, translation, okay, putting nucleotides together, putting amino acids together, other anabolic reactions. Well, I think that's really it, okay. I think there was some other stuff, but I can't remember it now. Check my paper. Glycogenesis, transcription, translation, yeah, and even things like DNA replication. You're making a whole new DNA molecule. That's going to take energy. Okay, other energy requiring processes that we should be familiar with, though at that time we might not really have thought about it in these terms. Movement, right? Movement. So we're talking about things like cell movement, cell shape changes. For example, phagocytosis takes energy to extend that membrane around pathogens, etc. Even muscle contraction. Okay, muscle contraction, that requires ATP as well. So we need to kind of have that general awareness of this idea, because you've done these things in other contexts, but when you do respiration, examiners could well kind of ask you to make that mental connection between the processes that you've looked at in other contexts and now through the lens of respiration that produces the ATP that is required for all those processes. Okay, so that's kind of why we need respiration. Now let's start looking at the process that creates ATP from glucose. Right, so we have two branches here. We have the part of respiration that happens in the cytoplasm, that's glycolysis. And in the mitochondria we have numerous stages, okay? So let's begin with glycolysis. And when we do anaerobic respiration, we're going to come back to this part. But yes, glycolysis. Now, glycolysis happens in the cytoplasm and it takes glucose as its main input. I'm just going to call it G, okay? Glucose is a G. Now, glucose, first thing that happens, remember glucose is 6 carbons, okay? So glucose, I mean, I like to simplify this process in 2 steps, even though there might be multiple ones. So first, we phosphorylate the glucose, so it becomes a double phosphorylated 6 carbon sugar, but then chopped in half, and at the middle point, what we end up with is 2 times 3 carbon sugar that's phosphorylated. 2 times, what do we call it? Do we call it a triose phosphate? So we have a 2 times 3 carbon sugar triose phosphate. That makes sense, triose phosphate, and that's each of the trioses is phosphorylated once. Okay, and that we have a 3 carbon. Now, to get that to happen, we do need the energy input. So we need to invest a little bit of energy to get this process started, and then it ultimately releases energy. Okay, so you could well ask, well, I thought this was a process about making ATP. Why are we using ATP? But it's just like many chemical reactions. You need to give them that activation energy before they overcome they get over the hill, and then they drop down much lower than when they started, releasing much more energy than you put in. Okay, so ATP to ADP, I believe we use 2 there, okay, to phosphorylate the glucose and get that process started. Then in the next stage, what happens is that each of the triose phosphate is converted to pyruvate. So the end of glycolysis produces 2 pyruvate molecules, which are both 3 carbon. Long story short, in that process, however, we net generate 4 ATPs. Okay, now this was not made in the mitochondria, so we call this substrate level phosphorylation. So I'm just going to add that term onto this glycolysis process, right? Substrate level phosphorylation, right? That's ATP produced, but it wasn't produced in the mitochondria. It wasn't produced by ATP synthase and all that stuff, which we will get to. So 4 ATPs produced, that gives us a net total of 2 ATPs produced. But at the same time, something also very important happens. We convert a coenzyme, NAD, and it gets reduced, so it becomes NADH. Okay? Again, we're not going to spend too much time going over the whole process. That there's other videos that do that job. This video is for revision purposes only. So and I believe the number here is 2. So for every 2 pyruvate molecules, we produce 2 reduced NADs, and a net production, so we lost 2 ATPs minus and plus, so that gives us a net plus 2 ATPs. So it's all the 2s. Just remember that number. Okay, 2 NADHs, 2 pyruvates, and 2 ATP molecules produced in glycolysis. Okay, that's fine. So things of importance. So we had a substrate level phosphorylation. Cool. We have reduced coenzymes. Now, it's easy to just kind of remember the NAD stuff. It gets reduced, fine. Okay. And we have begun the process of oxidation. So what we say is that glucose has been oxidized. Okay, so glucose is oxidized, but our coenzymes are being reduced. Okay, so glucose oxidized, coenzymes reduced. And that's really important, because if you know the process, it's the reduction, it's these coenzymes, because they're reduced, it's in their reduced form that they can fuel the electron transport chain and the proton gradient and the chemiosmosis and the ATP synthesis. So that's important in that context. And these ATP molecules are nice to have as a bonus. Okay, right. So now that cytoplasmic part of respiration is done, glycolysis is done, we then move on into the mitochondria. Before we get into it too much, I will just do a quick overview of the key structural elements that we need to know of the mitochondria, and then we'll get into where the reactions happen and so on. Okay, so mitochondria, first thing we look at is structure. Actually, do I even need to draw it? I guess I should. Okay, so the structure, we have an outer membrane, so an outer mitochondrial membrane. We have the inner membrane, which is highly folded to increase the surface area. Yeah, so we have an inner mitochondrial membrane. Inner mitochondrial membrane. Now the inner mitochondrial membrane is folded, and the structure, those folds are called cristae. Yeah, and the cristae have stalked, so you can observe that the cristae, they have these little bumps on them. Okay, now those stalked, they're called stalked particles, but essentially what they are is ATP synthase. They are ATP synthase. Okay, and we're gonna visit them anyway. Don't worry about that. Yeah, so the fold is important for surface area. The stalked particles are the ATP synthase. The inner membrane also has, the cristae also has the electron carrier proteins that form the electron transport chain, electron carriers, so those are important. And in the middle, the fluidic substance in the middle, that is our matrix, mitochondrial matrix. Yeah, and there will be circular DNA, there will be ribosomes around, and that is it, really. Okay. So let's get into it now. Let's get into the main part. So we will kind of continue this discussion. So we looked at cytoplasm and the processes that happen there. So the next thing we'll look at is the stroma. Next thing we'll look at is the matrix. So, because then the next important step that's going to happen is going to happen in the matrix. So now, once in the matrix, it's one reaction, but there's quite a lot of stuff happening in there. The link reaction is happening, okay? So I'm just going to label that here. Link reaction happening in the matrix, takes pyruvate, and importantly, what it does is, first thing, it is decarboxylated. So we've lost, we begin our process of breaking down the carbon skeleton of glucose. We've lost our carbon. Remember, 2 pyruvates would mean we lose 2 carbons as carbon dioxide. Right, so our decarboxylation process begins there. So that's an important thing in the link reaction. We have a decarboxylation. So that's important terminology. Again, we know these things are happening, but unless we say those things in an exam, even though we know it, it might not bear fruit. You know, the examiner needs to see that you know it, so you've got to use the right terms to describe what's going on. Okay, so in the link reaction, we have a decarboxylation, produce carbon dioxide. We also have a dehydrogenation. So that's not the first time that happened. Triose phosphate was dehydrogenated to produce the NADH. Okay, so again, we're going to have some NAD becoming NADH. So we have a decarboxylation. You can call this a reduction or a dehydrogenation. Yeah. Reduction right there. So remember though that the pyruvate is being oxidized. It's the coenzyme NAD that is getting reduced because it's gaining the hydrogen. Okay. Okay. Now this is the bit that I really would. It's a minor point. I don't think, this is a huge thing, but again, I don't want such things to be overlooked. So what happens is at the other side, you've got the 2 carbons of the the 2 leftover carbons from the pyruvate now. Those 2 carbons form a 2-carbon unit called acetic acid or acetate. And it is passed on to a coenzyme. So a coenzyme comes along and collects it. And that coenzyme that comes along is called coenzyme A. Okay, remember, coenzymes by themselves don't do anything, but they are very essential for other enzymes to do what they do. All right, now whichever enzyme it is that converts pyruvate to acetate, it cannot do it without coenzyme A being present. And at the end of it, it's coenzyme A that's left holding and carrying the acetate unit, the 2-carbon unit. So what comes out the other side is called acetyl coenzyme A. So it's important to understand that pyruvate is not converted into acetyl coenzyme A. Pyruvate is converted to acetyl, and it's CoA that is kind of carrying it to the next part of its journey in the Krebs cycle. Okay? Right then, so still in the matrix, so cytoplasm, so all this is happening in the matrix. So we've got the link reaction happening in the matrix. And then, continuing in the matrix, we have the acetyl-CoA then entering the Krebs cycle, or the tricarboxylic acid cycle. What happens then? Now, and remember that the acetyl unit is now, it's only 2 carbons because we lost some carbon dioxide in the link reaction. This continues to happen in the matrix of the mitochondria. So acetyl coenzyme A goes into the Krebs cycle, and what that essentially means is that the acetyl-CoA reacts, well actually, no, not the acetyl-CoA, but the acetyl unit is delivered. So the coenzyme A delivers the acetyl into this reaction with the 4-carbon oxaloacetate. Okay? And what happens is the 2-carbon of the acetyl combines with the 4-carbon of the oxaloacetate and produces the 6-carbon citrate. All right, and that's 6 carbons. And obviously, our coenzyme A will be regenerated because it's a coenzyme. It doesn't really take part in reactions. Well, it takes part in reactions, but it doesn't get changed by them. Okay, and off that coenzyme A goes to take part in the link reaction again. Meanwhile, what happens in the next stage of the Krebs cycle is that the citrate then gets converted into malate, I believe. And malate is 5 carbons, but to convert citrate into malate, which is 5 carbons, obviously we must have had, yes, another decarboxylation, right? So again, so now we're in the Krebs cycle. So Krebs cycle has its own key steps. So reaction number 1, right there. Reaction number 2, again, like I will label because we are revising, decarboxylation, important to remember. We also have more NAD reduction, so more NAD becomes NADH here, and then we produce malate. But again, just like in the Calvin-Benson cycle, you can't just have this process. You do need to regenerate, so that you do need to regenerate the oxaloacetate, because you can then keep the cycle going. Okay, so malate is then converted in back or used to regenerate oxaloacetate. A lot of stuff happens here. I believe we have a substrate level phosphorylation. ADP is converted to ATP. ADP is converted to ATP. I'm not going to focus on numbers here because I don't remember stuff off the top of my head like this. But also, as a revision exercise, I don't think this is essential right now. ADP, so we get a substrate level phosphorylation, but also we do get more NADH produced from NAD, so we got further reductions. But also another coenzyme gets involved here. An FAD is also converted to FADH. Okay, and I believe that is the main, or those are the main events of the Krebs cycle right there. Again, remember your reactions. Remember the steps that get produced: decarboxylation, reductions, coenzyme reduction, decarboxylation, and substrate level phosphorylation, and where they happen. Okay? Right, and that is essentially the stuff that happens in the matrix. Now, the next thing that we're going to look at is what happens to all this reduced coenzyme that has been produced, because this has been the point of the whole thing. Wait a minute, did I forget something? Yes, I did. That's 5 carbon, that's 4 carbon, so we must have had a decarboxylation as well, but not very conveniently, I don't have space to do that. So I'm just going to budge things up here. Malate, 5 carbons converted there, and yes, we do have the final decarboxylation, and that's 1 carbon, 2 carbon, 3 carbon, that's 1 pyruvate. For every 2 pyruvates, we're going to have 6 carbon dioxides lost, which means all the 6 carbons that we started off with glucose by this point are lost as CO2. Okay? Right, and then essentially the NAD and FAD story is what we follow at this point. Okay, and that happens in the inner mitochondrial, or at the inner mitochondrial membrane. Okay, so we could go like that. We're really following what's happening here with all those things. Okay, so yeah, the inner mitochondrial membrane is where the next stuff happens, and really the story starts at the electron carriers. Okay, the electron carriers, the components of the electron transport chain. Okay, what they do, remember, is that they receive the electrons in the form of the hydrogens. Okay, so the hydrogens are delivered essentially. So what can we say here? Really what's happening is NADH is splitting up here into NAD plus H. The H itself is splitting up into protons and electrons, and the electrons are kind of being delivered into the electron carriers. Okay, so the electron carriers, then they take these electrons, which it's only possible because of all the things that happened before, right? The electrons are then used, the energy of electron flow, so electron transport happens, electron transport, and thanks to electron transport, remember, because the electrons are being transported through the electron transport chain, the electron, just like in photosynthesis, the electron carriers are able to pump protons into the intermembrane space, which I forgot to label. So you've got the outer membrane and inner membrane, and in the middle you've got the intermembrane space. Okay, it's fine, it's not like a huge thing, but it's just a term that you can use to easily describe the space between the two membranes, and therefore, you know, not getting yourself muddled up in your explanation or having to find the right words at the time. So you've got electron carriers, then you've got electron transport, and because of electron transport we build a proton gradient. We build the proton gradient, and because we build the proton gradient, we can have, because of ATP synthase, the stalked particles on the inner membrane, and because we've got that gradient across the inner membrane, we have ATP synthase that can take advantage of that, and as a result of the process of osmosis, as a result of chemiosmosis, we can produce ATP. ADP to ATP. Okay, and that's the whole ball game. We make ATP at the end because of that. Okay, and this process is known, this whole thing is known as oxidative phosphorylation. Oxidative because the electron carriers are allowing the NAD to go from its reduced form to its normal form, or oxidized form if you want to call it that. So we're oxidizing the NAD, but we are phosphorylating. As a result of that, we're phosphorylating ADP to ATP. Okay, so oxidative phosphorylation. Cool. Now, where do the electrons go? So they are flowing, but they're not just kind of being chucked out. Electrons don't work like that. They really need a place to be carried. So the electrons go through the electron carriers, and at the end of the electron transport, the last electron carrier is cytochrome C, and it is cytochrome C that recombines or combines electrons with oxygen and protons. So the electron plus oxygen plus protons to form water, and that's the last remaining bit. So I mean that's really important, and I always forget it, but the oxygen that is needed in aerobic respiration is required to collect the electrons from the electron transport chain so that the electron flow can keep going, so that we can maintain the proton gradient, so that we can keep synthesizing ATP. Next, anaerobic respiration. So, with oxygen discussed, don't want to go into the detail of it right now, but without oxygen, the electron flow doesn't happen for the reasons that we've just discussed. If the electron flow doesn't happen, our coenzymes don't get oxidized. Remember oxidative phosphorylation. If the coenzymes don't get oxidized, every reaction that needed our oxidized coenzymes in the link reaction, in the Krebs cycle, and potentially in glycolysis as well, all of those reactions that needed the oxidized form of the coenzyme in order to accept the hydrogen, all of those steps wouldn't happen or couldn't happen, and therefore the coenzyme accumulates in the reduced form and everything kind of grinds to a halt. Okay, with that said, we can now discuss then anaerobic respiration, which occurs in the cytoplasm as well, so we go back to this part of it. Anaerobic respiration is simply a way to keep generating ATP, even though smaller amounts, right? Because oxidative phosphorylation generates a lot of ATP. In comparison, anaerobic respiration allows the temporary maintenance of ATP production in the absence of oxygen until the oxygen supply can be increased, right? So for example, if you're exercising, it takes a bit of time for your respiratory system to get upregulated to the point where you're breathing fast enough and your heart's beating fast enough to supply the oxygen needed for your new level of activity. So in between those 2 points, anaerobic respiration kind of keeps things kind of ticking over, all right? With the with certain undesired consequences. That's why it has to be temporary, which we'll discuss. So in anaerobic then, there is a pathway, or there are some fates of pyruvate that we can discuss. So, and it is to do with pyruvate, and it's all to do with how do we convert our NADH that we have back to NAD so that we can keep. Remember, this is the step that's making ATP right here. Okay, so how could we keep this reaction going when all our NAD is stuck in this NADH form? We need to find a way to get the NAD back. So remember, there's two kind of pathways. There are two things that can happen to pyruvate and we'll look at that right here. So in animals, pyruvate is converted to lactic acid, and in doing so, we get NAD, so we convert NADH back to NAD, right? And then that NAD can then be used to just run or allow that second step of glycolysis to carry on, keeping, 2 molecules of ATP made compared to around about 35, 36 by oxidative phosphorylation, but at least something is better than nothing. Okay, in the other thing that can happen is in yeast and plants. So in yeast and plants, the anaerobic respiration goes a slightly different way. The pyruvate is converted to alcohol, ethanol. Okay, but the same kind of thing occurs. We're taking NADH and we are returning it to its oxidized form. Okay, so there is, oh wait a minute, ethanol plus CO2. Right, yes, lactic acid is still 3 carbons, so we don't get any loss of CO2 there. But ethanol, that is a 2 carbon molecule. And so we have a decarboxylation step in that process as well. Okay, cool. So, yeah, and this is going to be then important when it comes to being able to measure anaerobic respiration. Because in yeast and plants, you could measure anaerobic respiration by the CO2 produced in the absence of oxygen. Right, okay. Finally then, let's go to the last bit, which is being able to measure respiration and the respiratory quotient, okay, which are kind of connected. Now, there are a number of ways that respiration can be measured. There's one main way which kind of demonstrates the whole principle very well, and I think all the other ways are kind of connected to this, and that is the respirometer. Respirometer. Very, very quickly, you have organisms in a tube, right? Organisms, you've got some organisms there, and they are respiring, and you've got a tube, a capillary tubing right there. Again, you've got another tube on the other side also. Okay, but this doesn't have organisms in it. This just has something that takes up the same mass. Now this tube is a capillary tubing, okay, and it's got a bit of liquid. And obviously, like you've seen that there will be something with like a ruler on it where you can measure the distance that the fluid moves. Maybe it shouldn't be there. Maybe that fluid would be better off somewhere else. So you measure the starting point and you measure the end point and you measure the distance in a given time so that you can work out the rate. But what the hell is actually going on here? So you got respiring organisms in one side, Okay, on the other side, you've got something, non-living, so it's not respiring, non-living. Okay, the idea being, this is all an airtight connection. So these organisms are respiring, and as they respire, they're producing CO2 and, taking in oxygen. So they're consuming oxygen, but evolving CO2. So overall, that wouldn't change the pressure in the tube, but right here, you've got something like potassium hydroxide or soda lime even, that absorbs CO2, and so the only thing that changes the pressure in the tube is the consumption of oxygen. So there's no CO2 that's actually being added, because the CO2 is going not in the gas in the tube, but it's going into the potassium hydroxide. So as this oxygen gets removed from the air in the tube and goes into the organisms, the pressure in the tube drops. The pressure in the other tube then becomes relatively higher, and therefore the fluid tends to move in that direction towards the respiring organisms. And if you measure the time and the distance that it moves, you can work out a rate of oxygen consumption. Okay? And really, then once you can do that, then you can investigate the effect of different factors, right? So you can treat these organisms in different ways according to the factor that you're investigating, keeping all other things constant, controlled, and then you can see how the changing of that factor affects the rate of oxygen consumption. Okay, and there are some other methods to measure respiration, but they're based on a similar principle. Okay, so you can change the conditions. For example, you can make those conditions anaerobic, and as long as you have something like yeast or plant material, in anaerobic conditions you can measure the CO2 evolved, and that will give you an anaerobic respiration rate, for example. Okay, so we have that, respirometer. Now, last thing in respiration is respiratory quotient, because different things can be used for respiration. It's not just glucose, right? For example, glucose can be used, so this idea that there are different respiratory substrates. Okay, so what can be used for respiration? Well, glucose is an example of a carbohydrate, right? And that would happen in the normal way. But you can also, for example, use lipids. Lipids can also be used for respiration. They don't really go through glycolysis, but lipids can be used to make acetyl coenzyme A. And acetyl coenzyme A can then just plug straight into the Krebs cycle, for example. Okay? And similarly, proteins may also be used. Okay, now the idea being that because these molecules enter the process at different points, carbohydrates are essentially the whole thing, lipids, for example, joining the process at the acetyl coenzyme A stage, because of that, the oxygen produced and the carbon dioxide evolved is different. Because if you have something coming into the process over here, we don't have that decarboxylation stage, do we? So when lipids are used, we wouldn't have as much CO2 produced by the process. Okay, and the same goes for proteins, because right now I can't remember how the proteins get used, but because the proteins would enter the process at a different stage, they would also have a different ratio of oxygen used for CO2 produced. Okay, and as a result of that, they all have different respiratory quotients. Respiratory quotient is the amount, or well, I shouldn't say, is it amount? Yeah, it might be amount of CO2 produced during the respiratory process divided by the amount of oxygen used. Okay? Now, carbohydrates have a respiratory quotient of 1. Okay? Lipids have a respiratory quotient of 0.7-ish. Okay? And proteins are at about 0.8. Nope. And proteins are at 0.9. So the kind of questions that might come up is, I don't think you're expected to remember that. It's usually that information is given to you, but based on that information and based on some experimental data, you might have to make a connection between, right, what's actually being used for respiration at this moment in time, given that the experimental data shows that the respiratory quotient is that, or that. Okay, and I think that is all I want to say about respiration. I hope that this has been useful. Do try and do this for yourself. It is kind of really, really useful. I know you don't see all the detail here, and I know we do tend to think of biology as all about the details, but I think you have a better chance of remembering those details if you do try and organize the information, the big picture like this, and the details will stick better as a consequence. The next one, energy transport in plants. The next one we'll do hormonal communication. Okay guys? Right, good luck.